Hi everyone, my name's David Nobbs. I work at Roche in the International Regulatory Policy team. I focus on digital health and data policy, but just recently actually moved over to this team after working for the last 5 years in developing digital endpoints for neurodevelopmental disorders. And I'm going to be talking about that work today, specifically the work we've been doing to develop novel endpoints for autism spectrum disorder using digital health technology. So as you may know, autism is a highly heterogeneous disorder, and it's heterogeneous both in terms of the different types of symptoms that individuals can have and their severity. So you can see associated symptoms mapped out here, including disruptions in sleep, mood and anxiety, and different individuals can have different aspects of those. So some individuals may have more problems with anxiety, others with more problems with, for example, aggression, and also the levels of severity. So it's very heterogeneous in that respect. But then you have the core symptoms of autism, or the core diagnostic criteria according to DSM-5, in these two dark blue circles in the middle. That includes social communication impairments, and that can include the way that people speak in terms of their expressive communication, also their receptive communication, and then also their social deficits, how they interact with people, do they avoid social situations, how do they behave during social situations. So actually even within one of those core criteria, there's a lot of heterogeneity of different types of social communication symptoms. And then the second core diagnostic criteria are restricted and repetitive behaviours, which again include quite a variety of different symptoms, including repetitive movements such as body rocking, hand flapping, running in circles, but also restricted interests, so focusing on the same topic and having a real deep interest in it, but to get to a point where they don't want to talk about any other topic and really become obsessed with it. And also liking to having a very structured way, structuring their day in a very ordered way or structuring a task in a very ordered way, to the extent that if you try to disrupt that, then they find that very distressing. So both of those two core diagnostic criteria actually encompass quite a lot of different aspects as well. Although this is a highly heterogeneous disorder, it's also a very widespread disorder, idiopathic ASD, and it impacts a wide part of the population. But there is no approved pharmacological treatment for these core symptoms. And so that's something which Roche has been concerned with and trying to develop new treatments for, and I will be coming to in a moment. So although there is no pharmacological treatment to treat the core symptoms of autism, there are behavioural interventions, cognitive and behavioural interventions, which have been used for that, but the trials that have been used for them are often underpowered and the evidence that they're effective in adults has been limited. There are pharmacological treatments for irritability in autism, so not for the core symptoms, but also those associated with side effects such as weight gain and sedation. So for this reason, Roche is trying to develop novel pharmaceutical treatments for the core symptoms of autism. One of the core challenges in doing that is to measure the effects of the treatment. So the typical way to measure the clinical benefit is to use an outcome assessment, either asking the participant to report on changes or to ask a caregiver to report on the patient's symptoms. But these can be associated with placebo effects, and there was a meta-analysis recently for ASD interventional trials which showed approximately 20% of participants were much improved with placebo. And you can actually see on this right plot here one of the Roche trials for balovaptim, where there was many participants appear to improve who were in the placebo arm. These placebo effects have been associated with the use of subjective caregiver-reported outcomes, and there's a lot to disentangle about why you might see that effect, but one reason could be because just participating in a clinical trial could alter the caregiver's perception of the symptoms. They may have a real hope for improvement, for example, and so then suggest that there has been improvement in later scoring of the same symptoms, rather than genuine improvements. So for this reason, we've been trying to develop objective measures of ASD symptoms using digital health technology. Why would we want to do that? Well, for one thing, when we assess participants in a clinic, we can only assess them very infrequently. And you may be assessing them on a particularly bad day or a good day, and that day may not then be reflective of their average symptoms around that period of time. But with digital health technology, they can take it home, they can use it on a daily basis, and we can get a better reflection of what their symptoms are like over a 2-3 week period, rather than just on a specific day in a clinic. We can also measure their symptoms using sensors which are high resolution. So a clinical scale will often just have 0 to 3 points on an ordinal scale. But with digital health technology, you can really have a high resolution scale for detecting improvements and differences between individuals. It can also be more reliable because you're using objective technology, which is likely to give you the same measure under the same conditions 1 week later, or more likely to do that than based on a subjective report. And for these reasons, we think that these objective measures are more likely to give us an accurate measure of treatment effects than a subjective evaluation, or at the very least, be complementary to that. Based on this rationale, we have developed a set of assessments that we call wavelength to use in our clinical trials. And this was originally informed by qualitative research with individuals with ASD and their families to understand what symptoms are most important to measure. We then developed some prototypes of the assessments, which we took to focus sessions in the UK and the US with individuals with ASD to get their feedback, and also took the input of experts through advisory boards and expert consultations, and then based on this developed our assessments. Here's an overall view of the different assessments, and they include active tasks which are completed on the smartphone for measuring working memory and social communication deficits, surveys which are also completed on the smartphone on a regular basis, which are similar to the surveys you would complete in a clinic but can be completed more frequently. And then a really quite broad range of different passive monitoring tools, which are either wearables or pieces of technology placed in the home for measuring the participant's symptoms without them needing to complete any task actively. I'm going to go through now a few of the active tasks to explain in detail how they work, to give you a feel for what we're doing. So the first task is called Find the Egg, and this is to measure spatial working memory. We know from previous research that individuals with ASD can have spatial working memory deficits, but we also know from FDA's Voice of the Patient, where they had meetings with individuals with ASD and their family to identify what's most important for them for developing treatments, and working memory deficits were indicated as significantly impacting the child's life. So we developed this task, which is a fun task on the smartphone, where the participant needs to look at these different chickens which are displayed on the screen and choose to tap one to see if it's laid an egg. And each of the chickens will lay one egg, and when they tap it, the chicken stands up and reveals that it's laid an egg. When they find an egg, they place it in a basket, and then they search for the next egg. Because each chicken will only lay one egg, they shouldn't go and search it again, they shouldn't tap on it again after they found an egg underneath it. That will count as an error. And also, when they've tapped on a chicken to see if it's laid an egg, they shouldn't go back and check that same chicken again. That would also then count as an error because they already have seen it hasn't laid an egg. And then based on this, we're able to look at their performance over different sizes of, so starting with 4 chickens and then increasing to 6 chickens, etc., up to 12 chickens. We're able to get an estimate of their working memory capacity, how well they're able to remember the location of the egg underneath the chickens. We can then use this in our clinical trials to see if we see improvements in their working memory capacity when they're under the pharmaceutical treatment. Second task is to measure emotional recognition. And we know this is important in autism, specifically also linked to everyday social functioning impairments. And there has been a task which is used in the academic literature called Reading the Mind in the Eyes test for measuring emotion recognition. And we developed a similar task for the smartphone. So the participant is shown faces of emotions, and the intensity of the emotion can vary and the type of emotion can vary. And they should then select which emotion they can see. When they correctly guess the emotion, then the intensity of the next face will be slightly less, meaning it's harder to detect what the emotion is because they're looking less happy or less angry. And when they get it wrong, it would then be a more intense emotion on the next go, meaning it would be slightly easier. Then we're able over time to find what their emotion recognition threshold is, where they're on average getting it 50% wrong or right on clinical trials, on different trials through the particular task. And with this emotion recognition threshold, we're then able to see if there are any treatment effects which modulate this emotion recognition threshold. The third and final task, which I'm going to talk about, is a relatively simple task for measuring expressive communication. So we know that expressive communication is part of one of the core diagnostic criteria of autism, and impairments can be reflected in how they speak in terms of how flat their speech is, how monotonic it is, but also do they take turns with different people in conversation, how much do they speak for, do they have long duration in their responses or short duration in their responses. These are different aspects that can be affected, and we know that in general conversation skills is a very important aspect to improve. So in fact it was the most frequently reported concept individuals with ASD in a family wish to improve. The task is very simple. The participant and their study partner, which could be for example one of their parents, should record a 5-minute conversation once per week. We are then developing automated methodologies for analysing this data, so it doesn't need to be listened to by researchers. And these methodologies include diarisation for working out who's speaking, and then automatic speech recognition algorithms for identifying what exactly is being said. And then we extract different measures from those automated analyses, including, for example, how long they speak for, how often they take a turn in a conversation, what's their pitch and their pitch variability, which is related to how they're expressing themselves. And then repetition, are they repeating the same words or phrases or the same topics in the conversation? So we're using those 3 and all of the other assessments in our different trials, and 2 of those are now completed, a phase 3 trial and also an observational trial. And we've been analysing those data, and we have results showing for those 3 tasks we've just discussed, strong results that showing correlations with existing clinical scales to support the convergent validity of those measures, and also differences between different known groups, and high test, retest reliability for our different measures. So now we are conducting 2 more clinical trials to validate those measures. And the first results will come out in the next 12 months from the first studies, and then we will be publishing our validation results showing we can replicate our findings. And then we hope that these will then go forward into further clinical trials to be used as endpoints. And that's really our overall objective, for these to be used as regulatory accepted clinical endpoints in studies to show treatment effects, to try to support the endpoints that we currently have and deliver improvements so we can really test the true treatment effects of our drugs that are being developed for autism. I'd just like to thank people that have contributed to the work. So first off, of course, the individuals with autism and their families, and some of those contributed to our focus sessions and helping us design the tasks. And then our collaborations within different functions of Roche, and then beyond Roche with the site investigators in our clinical trials, the Aims to Trials Consortium, and the advisory board members that contributed to the design. Thank you very much for listening to this presentation.